طيب اتفضل لو عايز تعمل مقدمة عشان توريهم الطريقة هتشتغل بيها بروف. شكرا جزيلا لاستجابتك لرغبة الطلبة ومشاركتك معنا يا بروفيسور طارق، إضافة متميزة جدا جدا. بارك الله فيك.
الله يسلمك دكتور عمار، وشكرا جزيلا على الدعوة، وشكرا جزيلا للطلاب اللي وصلوا وحضروا. ودي مجرد مراجعات يا شباب، يعني we are just doing revision as informed by Dr. Ammar that you already studied all this, and we are going just to revise, and I thought it will be better for you just to train you how to answer the multiple choice questions, the MCQs, better than to give you a new lecture. So we are going to discuss each topic, and we are going to very quickly, briefly, we are going to cover the most important information about each topic, and then we are going to practice immediately the questions about that topic. Just to tell me, any student who can just tell me about which systems already covered until now? Yes, Shabab, active participation is better. If you can just tell me which systems until now you have covered. Feel free, please, and open your mic and just start to talk.
We covered most of the systems. We just have, we're halfway through CNS and reproductive, but we finished the rest.
Okay, thank you. So even the CNS, you started already the CNS, the sensory system, motor system, you have covered?
We did the motor system and the special sense. Yeah, I think sensory
too.
too.
Special sense is also good. Okay, okay. So already you are towards the end. Very good. Okay. So we are starting electrophysiology from the beginning. This is just an introduction. I just want to go through the excitable tissues. Through the excitable tissues, I will just try to know to which extent you understand physiology, and accordingly then we can go into the sensory system and motor system and so on. So let's start. Are you ready to start? Yes, you can answer. Please don't wait until I give you a chance. You can just give your answer immediately. If you are ready to start, we are going to start now. Right. I will start with just revision of the channels, cell membrane channels. Very briefly, I will go through that, and then we are going to answer question number 1. There are 3 types of membrane channels. In brief, we have voltage.
بروفيسور طارق معلش للمقاطعة، أنا أونكولو في المستشفى، فلو بقيت اوف شويه معناه بتكلم معاهم او نادوني او كده، ممكن تواصل عادي يعني في المحاضره.
خير، ما في مشكله ان شاء
الله. المشكله بتكون في الصفحات، لكن بحاول انه ما اكون غايب كثير.
خير ان شاء الله، براحتك يا عمار ان شاء الله. شكرا جزيلا. Okay, so we have 3 types of channels. We have voltage gated channels, we have ligand gated channels, and we have leak channels. Leak channels are leaky. They are always open. So ions can go in and out through these channels. We have leak channels for sodium and leak channels for potassium. So, as you know, potassium is more inside than outside. And sodium is more outside the cell than inside the cell. So, through the leak channels, potassium always go out. There is potassium efflux. And sodium, through the leak channels, always go in. There is sodium influx. So there is influx for sodium, tendency to influx for sodium, and tendency to efflux for potassium. But the cell membrane is permeable to potassium more than sodium. So potassium will go out down the concentration gradient through the leak channels. But sodium cannot go in because the hydrated atom of sodium is large, so it cannot go through the leak channels, and the leak channels are guarded by calcium. So calcium is closing the leak channels for sodium. So again, For the leak channels, these are the needed information. Leak channels are found in all cells of the body. Leak channels are always open. Leak channels for potassium will allow potassium to go out. Leak channels for sodium should allow sodium to go in, but sodium cannot go in because the hydrated atom of sodium is bigger, and the leak channels are guarded by calcium ions. So during hypocalcemia, calcium can get in, but whenever calcium is normal, sodium cannot get in. During hypocalcemia, I mean sodium can get in, but whenever calcium is normal, sodium cannot get in because calcium guards sodium channels. Okay, so this is the needed information about leak channels. We have voltage gated channels, and these will open and close according to the change in voltage, according to the action potential itself. If there is change in voltage, then the leak channels will open and then close. We have voltage gated for sodium and we have voltage gated for potassium, but they are found only in excitable tissues, in nerve and muscle, and they are responsible for the action potential. That's why action potential occurs only in nerve and muscle, because it needs the voltage gated channels for sodium and for potassium. And we have ligand gated channels, and these channels will open and close whenever there is neurotransmitter or hormone binds to the channel. So some of these channels will act as receptors, or the receptor can be associated with the channel. For example, a hormone will bind to the receptor and then the channel will open, and then sodium can get in, or potassium can go out, or other ion also can move. So these are the three types of channels. Ligand gated channels can open and close, due to binding of a hormone or neurotransmitter. Voltage gated channels can open or close according to the change in voltage, and leak channels are always open. Okay, so as I mentioned, the voltage gated channel is responsible for the action potential, but the leak channels are needed for the resting membrane potential, because the resting membrane potential has three causes. If you remember, unfortunately, you are not eager to answer, so I am asking the question and answering it immediately. Okay, so we have three causes of resting membrane potential. Number one, potassium efflux through the leak channels. Potassium efflux, that means potassium will go out from inside to outside through the leak channels, is the first cause and the most important cause. More than 90% of the resting membrane potential is due to potassium efflux. But sodium cannot get in, as I mentioned, only potassium efflux. And for each potassium going out, negative charge inside the cell is generated. That is how the resting membrane potential is generated. Negative charge is generated inside by potassium efflux. More than 90% is due to potassium efflux. That's one. Second is sodium-potassium bump, because it can take 3 sodium to outside and 2 potassium to inside. By doing this, it will generate a negative charge inside, and this negative charge will contribute to the generation of resting membrane potential, but it is a minor cause. And a third cause is the non-diffusible anions like phosphate and protein inside the cell. This also will add the negative charge inside, and this will generate the resting membrane potential. So now we go to the question number 1. The resting membrane potential is generated by, have a look and very quickly try to select the answer. I will give you 1 minute for each question. Okay. After my explanation, you have to select the answer. Then after that we can. Okay. Now the resting membrane potential is generated by opening and closure of ligand-gated channels. Okay, this is incorrect. Okay, because ligand-gated channels are not needed for the resting membrane potential. What is needed is the As you remember, what is needed is the leak channels. Passage of ions through the voltage-gated channels. Voltage-gated channels are not needed. Selective permeability of the cell membrane to potassium. This is what? This is the leak channels. This is the correct answer. Closure of sodium leak channels by calcium. As we mentioned, this will prevent sodium to get in, so it is incorrect. And the high concentration of phosphate inside the cell, this will contribute to the resting membrane potential, but as I mentioned, this is a minor cause. This is not the major cause of the resting membrane potential. Okay, the major cause is the, as I mentioned, the major cause is the potassium efflux through the cell membrane, and the cell membrane is permeable to potassium. Okay, we go to the second question about the action potential. As you remember, the action potential consists of depolarization and repolarization. Depolarization is due to sodium influx, and repolarization is due to potassium efflux, and usually action potential is generated after sufficient stimulation. So the definition of action potential is that it is a reversal of membrane polarity after sufficient stimulation. Sufficient stimulation is needed to raise the resting membrane potential to the threshold. And then depolarization and repolarization will occur. Okay, let's go through the options and then we are going to select the answer. An external stimulus is not always needed for its initiation. As I mentioned, external stimulus is needed for action potential, but not always, because sometimes action potential can be generated by automaticity. In the cardiovascular system, you know, the conductive system can generate action potential spontaneously when the conductive system, through the pre-potential, elevates the resting to the threshold. This is another story. Okay, but this is correct, not always needed. So this is the correct answer. The depolarization phase is always due to sodium influx. Some action potentials are generated by potassium influx. Can you mention in which cells depolarization is caused by potassium influx rather than sodium influx? Potassium influx, yes. Okay? Potassium influx is the cause of the depolarization. In which cells? Anyone?
في الاير يا دكتور، حاجة في الاير زي
Excellent. Yes, في ear, في inner ear. Yes, في hair cells. ممتاز. Okay, that's correct. So in the hair cells, depolarization is due to potassium influx. Okay, this is the only side. And in the cardiac muscle also, in the conductive system, depolarization is due to calcium influx in the conductive system. So potassium influx in the hair cells and calcium influx in the conductive system, that means sodium influx is not always the cause of depolarization. Its duration is equal in all types of excitable cells. طبعا this is incorrect because in the skeletal muscle the duration of the action potential is 20 milliseconds. In the cardiac muscle it is 400 milliseconds because of the what prolongs the action potential in cardiac muscle? What makes the cardiac muscle action potential very prolonged?
The plateau phase, because of the calcium.
The plateau phase. So the plateau phase due to calcium influx. After depolarization phase is due to activity of the sodium potassium pump. This is incorrect. After depolarization is due to extended potassium efflux. Okay, because in the repolarization phase, action potential, usually there is potassium efflux, and this is extended. It will continue more, resulting in the after depolarization phase. Then after hyperpolarization is by sodium potassium pump. So remember, depolarization is due to sodium influx, repolarization is due to potassium efflux. After depolarization is due to more efflux of potassium, extended potassium efflux, and then after hyperpolarization is due to sodium potassium pump, and it can be summated in skeletal muscle but not the cardiac muscle. Also incorrect, because summation occurs for the local potential, not the action potential. Okay, we have three differences between the local potential and the action potential. Local potential is the electrical activity that occur locally in the receptor, like receptor potential, or in the synapse, like excitatory postsynaptic potential. These are known as local potentials. So before generation of action potentials, we have local potential. In the receptor, usually there is local potential known as receptor potential or generator potential, and this should be summated to give action potential. Excitatory postsynaptic potential in the synapse, multiple excitatory postsynaptic potentials can be summated by spatial summation to give action potential, or by temporal summation to give action potential. But action potential cannot be summated. This is one difference. The second difference is that local potential is graded, can increase and decrease, but action potential is all or none. So local potential can be summated, and local potential is graded, but action potential is all or none. And as I mentioned, local potential can be generated by sub-threshold stimulus, but action potential is generated by threshold or supra-threshold stimulus. Okay, and these are the differences between the local potential and the action potential. Third question concerning myelination. Myelination, as you know, is formed by the cell membrane of Schwann cells and oligodendroglial cells. Oligodendroglial cells will form myelination in the CNS. Schwann cells will form myelination in the peripheral nervous system. And the myelin sheath is the cell membrane. That means it is phospholipid plus protein. So because it is the cell membrane of Schwann cells or the cell membrane of oligodendroglial cells. That's about the myelination. This is the information you have to know about the myelination. So let's start to answer the question. Concerning myelination, which is the following is not true. Not true, so we are asked, we have to select the incorrect answer. It is synthesized from phospholipid and protein. This is correct because it is the cell membrane. Okay, and it is found in preganglionic sympathetic neuron, and it is found in postganglionic sympathetic neuron. So this depends on the types of neurons. You know the types of neurons? We have type A, type B, and type C. Type A thick myelinated, type B thin myelinated, type C non-myelinated. Okay, so which of these is non-myelinated type C? The preganglionic sympathetic or the postganglionic sympathetic? I will make it easier for you. The preganglionic sympathetic is type B. The postganglionic sympathetic is type C. Okay, so the answer will be C, found in postganglionic sympathetic neurons. This is incorrect because there is no myelination in type C neurons. Okay, in the postganglionic sympathetic neurons. So it is easy to remember. The preganglionic type B, the postganglionic type C in the sympathetic and the parasympathetic neurons. Definitely myelination is found in the CNS, and it is formed by oligodendroglial cells. So we finished question number 3. We got question number 4, and this needs understanding of resting membrane potential. If ECF potassium concentration is increased from 3.5 to 5, number 1, you have to remember 3.5 to 5 within the normal limits, because the potassium concentration is from 3.5 to 5. So now potassium is increased within normal limits. This is not abnormal. This is normal limits. Okay? That's number 1. Okay? And if you remember, okay, if you remember, potassium efflux is the cause of the resting membrane.
Can you please all make sure your mics are muted? Sometimes people open the mics accidentally.
Yes. Okay. So we have, as you can see, from 3.5 to 5, this is within normal limits. And if you remember, potassium efflux is the cause of the resting membrane potential. Now potassium is going out. Okay, it's going out, and for each potassium ion going out, there is generation of negative charge inside. There is generation of resting membrane potential. So potassium will go out until minus 90 millivolt is generated. This is the resting membrane potential. If potassium outside is increased, that means the efflux of potassium is going out, is decreased. So we cannot reach minus 90 millivolt. We are going to reach, for example, minus 80 millivolt, because potassium outside more will prevent more efflux of potassium. Okay, so now we have less negative charge inside. Now let's go to the options. The resting membrane potential becomes less negative. This is the correct answer from the beginning. That's the correct answer. The resting membrane potential will not change. It will change definitely from minus 90. It will become, say, minus 80. Fast voltage gated sodium channels will open. It is not related. Action potential will occur spontaneously. Yes, action potential can occur spontaneously if potassium is increased more than 5. Because now potassium is increased 3.5 to 5. If it is increased up to 6, action potential will occur spontaneously, because the resting membrane potential will be from minus 90 will become minus 80 or minus 85. And this is the threshold, minus 75. This is the threshold. So action potential can occur spontaneously if it is 6 millivolt, not 5 millivolt. And the cell membrane becomes hyperpolarized. This is opposite. It is depolarized, not hyperpolarized. So the correct answer is answer number A. We go to the leak channels. Already we described the leak channels. These are the options. I will give you 1 minute to find the correct answer.
You have to interact in the lecture. When Professor Tarig asks a question, feel free when he asks you for the answer to unmute yourself. لأنه في delay في الشبكة يا أخواننا، خليكم سريعين شوية. If you know the answer, just unmute yourself and say the answer.
Okay, sometimes the students are shy, so no problem. So leak channels, the correct answer is answer number A, are present in almost all cells of the body. We mentioned that leak channels are found in all cells of the body, but voltage gated channels are found only in excitable tissues, nerve, and muscle. Have gates that open and close during action potentials. During action potentials, voltage gated channels are activated, but leak channels are not activated. Have gates that open when a hormone binds to the receptor. This is the ligand gated channels. We described that at the beginning. Are responsible for depolarization phase. These are the voltage gated channels for sodium. Gates are closed by the end of the action potential, not related. So the correct answer is answer number A. We can go to question number 6. Calcium channels in the synaptic node. So very quickly we are going to describe the synaptic transmission. The steps of the synaptic transmission you have to know. First step is arrival of action potential to the terminal end of the axon, and the second step is opening of calcium channels. These calcium channels will open, and then calcium will get inside. This will increase movement of the vesicles in the synaptic node, and by exocytosis the neurotransmitter in the vesicles will be released to the synaptic cleft. After that there is binding of the neurotransmitter with the receptor and generation of excitatory postsynaptic potential followed by action potential. These are the steps. Again, I am going to mention them. First step, arrival of action potential to the synapse. Second step, opening of calcium channels, allowing calcium influx. Third step, increase movement of the vesicles and release of the neurotransmitter by exocytosis. Then after that, the neurotransmitter will bind to the receptor and generates excitatory postsynaptic potential. Then after that, there is summation of excitatory postsynaptic potential to generate action potential. These are the steps of the synaptic transmission. So now we are talking about the calcium channels, the second step. Calcium channels allow release of neurotransmitters through them? Definitely no. Allow only calcium influx. Are voltage gated? As we mentioned, they open after arrival of the action potential. So they are voltage gated. Yes, this is the correct answer. Ligand gated? No. And are opened when the acetylcholine is released? Also no. And cause termination of the action potential? Not related. So the correct answer is answer number B. This is like that. We go to the proteins involved in skeletal muscle contraction. All these are involved in skeletal muscle contraction except calmodulin. Calmodulin is the calcium binding protein in smooth muscle. So in smooth muscle, the protein that binds calcium is calmodulin. In skeletal muscle, the protein that binds calcium is troponin C. So you have to remember that, and these questions usually are repeated. So when you know the answer, I think whenever this question you find it again, immediately you can answer it. Calmodulin, calcium binding protein in smooth muscle. Troponin C, calcium binding protein in skeletal and cardiac muscle. The A band in striated muscle, we have two bands, A and I. I is isotropic to polarized light. Iso means the same. That means the type of protein is the same, which is actin. So we have actin in iso, in the I band. A band, anisotropic, that means there are different colors or different types of proteins which are actin and myosin. So the A band contains both actin and myosin, and it is dark because myosin is thick. That's why it is dark. But I is light because actin is thin. So very easy. I only actin, A, actin and myosin. So these are the options. A band is a light area. No, it is a dark area. It contains thick filaments only. No, it contains thick filaments plus thin filaments, actin and myosin. Divided by the M line, that's the correct answer. It is divided by the M line. Is longer during relaxation than during contraction. This is very important information. Usually the A band remains constant during contraction and relaxation. I band will decrease in size because of the overlap between actin and myosin. And appears due to overlapping between actin and myosin? No, it is appears because of the thick filaments only. So the correct answer is answer number C. We go to question number 9. Concerning chemical conduction, which of the following is not true? Already we described the synaptic transmission, but we didn't describe the excitatory and inhibitory postsynaptic potential. Usually after binding of the neurotransmitter with the receptor, many types of channels can open. Sodium, and this will allow sodium influx. That means there is depolarization. Potassium will allow potassium efflux. That means there is hyperpolarization. So when sodium gets in, causing depolarization, this will cause excitation. It will result in excitatory postsynaptic potential. When potassium channels are open, allowing potassium efflux, this will cause inhibition, resulting in inhibitory postsynaptic potential. If chloride channels are open, chloride will go in or out. This depends on chloride higher in the extracellular fluid or intracellular fluid. You know, chloride is high in ECF, extracellular fluid. So there is chloride influx, and it is negatively charged. That means it will increase the negative charge inside, resulting in hyperpolarization and inhibition. So chloride influx will cause inhibition. Potassium efflux will cause inhibition. Sodium influx will cause excitation. Calcium influx will cause excitation. Again, I am going to repeat it again because it is very important, and usually questions will come here. Excitation, when the neurotransmitter binds to the receptor, there are two options: excitation or inhibition. Excitation occurs by sodium influx or calcium influx. Inhibition occurs by chloride influx or potassium efflux. Okay, so these are excitation inhibition. So the question is about concerning chemical conduction, which of the following is not true? Is unidirectional, one way? Definitely it is one way direction from the pre-synaptic to the post-synaptic. Excitatory neurotransmitter open sodium channels? Yes, that's correct. Excitation by sodium. Inhibitory neurotransmitter open calcium channels? No, calcium also will cause excitation, so this is incorrect. GABA opens chloride channels. Yes, GABA is inhibitory neurotransmitter, and usually it acts by opening chloride channels. So that's correct. And the postsynaptic neuron is influenced by many presynaptic neurons. Usually the postsynaptic membrane is influenced by many presynaptic neurons. Yes, correct. So the correct answer, the incorrect answer, I mean C. Okay, so C is a selected answer. Now we come to ryanodine receptors. Ryanodine receptors and dihydropyridine receptors. These are the receptors involved in muscle contraction. As you remember, for muscle contraction, there is generation of the action potential at the neuromuscular junction. Then the action potential is transmitted through the sarcolemma. So through the sarcolemma, action potential is conducted. And it is conducted to the interior of the cell by the T-tubule. You remember the T-tubule? T-tubule function is to transmit action potential from the cell membrane, from the sarcolemma, to the inside of the cell. When the action potential passes through the T-tubule, usually it will open dihydropyridine receptor. So we have dihydropyridine receptor first, and then ryanodine receptor second. Dihydropyridine receptor usually is found in the T-tubule. And usually there are two different functions for dihydropyridine receptor, in skeletal muscle and in cardiac muscle. In cardiac muscle, dihydropyridine receptor will open, allowing calcium influx. That's why calcium from outside will participate in muscle contraction in cardiac muscle. But in skeletal muscle, dihydropyridine receptor will detect the arrival of action potential and will induce the opening of ryanodine receptor. So dihydropyridine receptors act as sensors for the action potential in the skeletal muscle. Okay, and after that, ryanodine receptors accordingly will open to allow release of stored calcium from the sarcoplasmic cisternae to the sarcoplasm. So again, there are two types of channels. We have the receptors, dihydropyridine receptors and ryanodine receptors. Dihydropyridine receptors comes first in the T-tubule, act as sensors or allow calcium influx, depending on whether it is skeletal or cardiac muscle. Ryanodine receptors are found in the sarcoplasmic cisternae, and they allow release of calcium, stored calcium, from the sarcoplasmic cisternae into the sarcoplasm. Okay, so now let's go to the questions. Ryanodine receptors allow influx of calcium from ECF to ICF in cardiac muscle? No, these are the dihydropyridine receptors. Allow release of calcium from the sarcoplasmic cisternae to the ICF in skeletal muscle? Correct, this is the function of the ryanodine receptor. Found in skeletal muscle but not cardiac muscle? This is incorrect. Act as sensors for arrival of excitation through the T-tubule. These are the dihydropyridine receptor. Allow bumping back of calcium to sarcoplasmic cisternae? No, this calcium magnesium antiport, which is activated during muscle relaxation. Last question about isometric and isotonic contraction, and it is very easy. Isometric means the same length, and isotonic means the same tension. The same tension means the tension is the same. There is no increase or decrease in the tension. But in isometric contraction, the length is the same, but the tension is not the same. The tension is increasing. Usually, isometric contraction occurs when you try to lift very heavy object. So, starting to lift very heavy object, you fail because you cannot lift it. Then you are going to increase your tension. That's why isometric contraction is associated with increase in tension. In isometric contraction, you elevate anything with the same tension, a light object will be elevated up. So it is the same tension, but the muscle will shorten. So these are the differences between the two. Isometric contraction differs from isotonic contraction. It does not occur in cardiac muscle. Both occurs in two types of muscle, so it can occur. It occurs during walking. During walking, usually you can use isometric and isotonic. Both of them are occurs. Okay. During standing, you are using isometric only. Okay, standing isometric, but walking is isometric and isotonic. It does not involve interaction between actin and myosin. It involves interaction between actin and myosin definitely, because it is a muscle contraction at the end. The muscle tension applied is increased during contraction. Exactly, in isometric contraction this occurs. The muscle tension will increase. In isotonic, the same tension is used. The force applied equals 0, the work is equal 0, but the force will be applied. So that's incorrect. So the correct answer is D. Yeah, that's all. By doing this, we revise the excitable tissues. Next time, inshallah, we are going to revise the sensory or the motor system. What do you mean? You finished the sensory system as well?
Yes.
Can we start with the sensory system next time?
Yes, we've already finished it, so we can start it next time.
Okay, so sensory system next time, and followed by special senses, and after that we can start the motor system. Okay, into lectures, inshallah. So see you, inshallah, next Friday, 9 o'clock. Sensory system MCQs. Better if you can read before you come, but even that we are going to discuss before each question. No problem. Okay. Any question?
شكرا جزيلا بروفيسور طارق. قبل الأسئلة، في طريقة نخط تايم تيبل للمحاضرات دي على أساس إنه التحضير يكون للمرات الجاية أفضل، الطلبة يكونوا قارين عشان يقدروا يعملوا انتراكشن أكتر ويجاوبوا على الأسئلة.
بالضبط.
حس البلان بدأت أوضح.
السنسوري سيستم اظنه يا عمار لو هم وافقوا على السنسوري سيستم اظنه لو بيجوا جاهزين للسنسوري سيستم واضح بالنسبه لهم.
تمام، يعني المحاضرة الجاية حتكون على السنسوري سيستم.
ايوه، بعدها ممكن تكون السبيشال سنسس على حسب الكتاب. ايوه، زي ما الكتاب ماشي يعني.
تمام، يعني كل شابتر تقريبا من الشابترز دي بمحاضرة.
ايوه ان شاء الله ان شاء الله.
تمام. في أي أسئلة أو تعليق على المحاضرة من الطلبة؟ تفضلوا.
يا بس عندي سؤال عايز أسأل منه بروف في حتة الإكزايتيشن بتاعت النيورو ترانسميتر. هو قال صوديوم كالسيوم بوتاسيوم and كلورايد، فبس عايز في حتة الصوديوم قال انفلكس ولا انفلكس ولا الحاجات دي ممكن نعيدها تاني؟
جدا جدا. طبعا انت عارف انه الصوديوم اكتر outside than inside، يعني الصوديوم خارج الخلية أكثر من داخل الخلية، عشان كده ما عنده حل غير انفلكس، إنه يدخل من أعلى لأقل. البوتاسيوم داخل الخلية أعلى من خارج الخلية، يبقى ما عنده حل غير إنه يخرج إيفلكس، يطلع لبرا. فعشان كده لما الصوديوم يدخل لجوا ده بيعمل اكزايتيشن، ولما البوتاسيوم يطلع لبرا بيعمل انهبيشن، طبعا لأنهم الشحنة بتاعتهم موجبة. بالنسبة للكلورايد لأنه سالب ولأنه خارج الخلية أعلى من داخل الخلية ما عنده حل إلا إنه يدخل، فكلورايد انفلكس، فده بيعمل انهبيشن لأنه حيزيد النيجاتيف تشارج انسايد، بيعمل هايبر بولاريزيشن وانهبيشن. بالنسبة للكالسيوم زي الصوديوم، هو أعلى خارج الخلية من داخل الخلية، يبقى حيعمل انفلكس وبالتالي بيعمل اكزايتيشن. فعشان كده مرة ثانية الاكزايتيشن هو influx of sodium, influx of calcium, inhibition, efflux of potassium or influx of chloride.
اوكي يا دكتور. تاني بالنسبة للجلوتامات بيعمل اكزايتيشن صح؟
ايوه، يبقى بالتالي حيكون بيستخدم يا الكالسيوم يا الصوديوم على حسب التقسيمة بتاعتنا.
اوكي. تاني عايز اضيف اضافة بالنسبة للريفيجن. نحن انتهينا الموتر. في السنسري ادونا اخر حاجة السيربيلم، والسبيشال سنس بدوا لنا في الاي بس، يعني كان
يعني ادوكم الريسبتورز والسنسوري تراكس ولا لا؟
ادونا التراك في الموتر انتهيناه كله.
لكن سنسوري تراكس مدوكم له الريسبتورز والبين
الريسبتورز والريفلكس والكلام ده ادونا ليه. البوستر لسه ما ادونا ليه.
مم. ايوه، انا ما
آه.
قصدي دايما احنا في النيرفس سيستم بنبدا بالسنسوري سيستم، ريسبتورز وبين وتاتش سensation.
دي
فده
الجونة ليه؟
فعشان كده احنا هنقوم نبدا بالسنسوري سيستم اللي هو البين والتاتش سensation والسنسوري ريسبتور والسنسوري تراكس.
اوكي.
بعد كده عندنا الفيجن والهيرنج. بعد كده عندنا الموتور سيستم اللي هو الريفلكسز، الاسترتش ريفلكس والانفيرس ستريتش ريفلكس والموتور تراكس. بعد كده بايراميدال سيستم والاكسترا بايراميدال سيستم اللي هو البيزل جانجليا والسيريبيلا. حتى بعد كده اخر حاجه اللي هي الهاير فانكشنز اللي هي الميموري والسبيتش. فانتوا بيكون باقي لكم الهاير فانكشنز، طالما وصلتوا لحد السيريبيلا معناه خلصتوا تقريبا، ما فضل لكم الا الهاير فانكشنز اونلي.
اوكي.
ف سنسوري سيستم، ولو لسه لو مفتكرين ان السنسوري سيستم ممكن يتاخر شويه، ما عندي مانع يعني، بس في الجروب ذاته ممكن تدوني هنت انه بدل السنسوري سيستم نحول لسبيشال سنس او نحول لموتور سيستم، ما في مانع، ما في مشكله.
يا دكتور السلام عليكم، عفوا.
ده كلام ممتاز جدا جدا، جزاك الله ألف خير يا دكتور طارق، وإن شاء الله في ميزان حسناتك. المحاضرات دي كلها بتترفع في اليوتيوب ونحضروها باقي الطلبة. اليوم كان في انقطاع في التيار الكهربائي والإنترنت في المدينة بتاعتهم في تيجال، عشان كده الحضور كان فيه مشكلة، لكن بقي الطلبة يحضروا المحاضرات. والمرة الجاية نوعدك نيابة عن الطلبة إنه التحضير يكون أفضل، وكل الأسئلة يتم إجابتها عن طريق الطلبة إن شاء الله.
خير ان شاء الله، ما في مشكله يا عمار. انا متعود على الطلاب ما بحبوا يشاركوا او عدد قليل منهم بيشارك اثناء فلو شاركوا الحمد لله، ولو ما شاركوا ما مشكله طالما هم بيستمعوا كويس، وان شاء الله بس يكونوا مستفيدين. وطالما برضو التسجيل موجود، الناس اللي ما حضروا بيكون موجود عندهم. والناس اللي بيحضروا ديل يعني هم يعني فيهم يعني هم يعني جايين برغبه. وانا يعني دائما بكون سعيد انه حتى لو في عدد قليل من الطلاب انه يستفيدوا يعني. فشكرا لهم على حضورهم، وشكرا لك على تعبك، وإن شاء الله إن شاء الله يكون في فائدة بس في الحاجة اللي بنقدمها.
ربنا يديك العافية يا أخي كثير. شكرا جزيلا على المشاركة.
الله
الطلبة
يبارك فيك.